So finally, I would like to show you some of the proteogenomics works that we've been doing, especially with regards to alternative splicing. So alternative splicing is a process in which exons of RNA recombine in multiple ways during pre-mRNA splicing. And once the pre-mRNA is spliced, it forms a mature mRNA, which is sent out of the nucleus into the ribosome where it produces the protein. So splicing can happen in many ways. And these are some of the examples that I've shown where you can splice out the introns in different ways. In a typical case where there is no splicing, where there is no alternative splicing, natural splicing occurs to form this cassette exon where you incorporate all the 3 different exons. But it can also skip certain exons, and that alternative splicing is called exon skipping. And 80% of the alternative splice isoforms are due to this type of exon skipping. There could be also cases where the branching point of the splice site can move. In this case, it moved towards the 3' end in the second exon and formed alternative 3' splice site, and that's called the alternative 3' site selection. And it could also happen that this exon moved towards the 5' end to form a new 5' start site. There could also be cases where the entire intron could be retained. This has happened especially in the cases where you find smaller introns, and we've seen those examples as well. And there's also possibility where exons can mutually exclusive. And this I've shown before in the FGFR2 receptor, where mutually exclusive exons are present. There could be alternative promoters and alternative poly-A tails. So all these different splice isoforms can lead to health and disease. In the cases of disease, in what we are concerned is breast cancer, the differences in alternative splicing, in this case, where you see a frame shift to form a small isoform due to these mutations, could lead to this breast cancer. And there have been a number of genes that have been already annotated to be involved in splicing and involved especially in breast cancer. So our approach is to look at all unannotated spectra to find out if there are alternative possibilities or alternative assignments to our spectra that did not get really assigned in our database searches. So we do that by producing a database consisting of all possible exon-exon combinations, exon-intron combinations, and intron-exon combinations that could give us a chance of assigning our spectra to a new splice boundary that is not annotated in UniProt. So we also obtain RNA-seq data for both basal and luminal cell lines as well as our tumors, and we run them through this program called MapSplice, which gets all these reads, RNA reads, and maps them to a reference genome and finds out the possible splice sites, both theoretical splice sites as well as splice sites that are already been annotated. Then we collect both these data sets and try to identify new isoforms, first using each data set to verify each other, and then finally visualize them using a UCSC genome browser. So this part of the talk, most people may not be familiar of the genome browsers that we use. This is the UCSC genome browser. So to give you an introduction of what this tells is the tracks on top are actually our peptides that we've identified. So these are bed files or bed tracks, data tracks that we assign to the genomic loci. So the bottom half shows the actual gene, various isoforms of the actual gene to which it was assigned to. So the dark lines are the exons. The thin lines connecting them are introns, which are the non-coding regions. And at the bottom, you see is a splice graph, which shows all the possible exon-exon combinations that one could have to give rise to all the different spliced isoforms at the transcript level and as well as the protein level. So if you look at a specific peptide, which I marked here, and expand or zoom in on that region, you can see that these two particular peptides that we've identified span this splice boundary. So half of the peptide sequence is identified or assigned to this exon, and the other half is assigned to this exon. So this could only be possible if there is splicing of this intron out. And we also see a bunch of other peptides surrounding both these exons, which give us extra evidence that this could be a possibility. What is also interesting is that we were able to see them in duplicate runs, and most of the splicing that I'll be showing are by replicate analysis and in both cell lines or both tissue types. And we can see that in this particular case, we see a miscleavage, mistriptic site here, which gives us extra confidence the presence of this splice site. So from now on, I'll show you a few examples of various types of splicing that we've identified, and in some cases, we are able to also do quantification on these splice sites. So this is an example where the peptide was actually a retained intron. So in this gene, this peptide was actually assigned to the beginning of this gene, and if you zoom in on this region, you can see that these peptides that are shown in red in both runs actually span this exon-intron boundary. So originally we thought that these peptides were actually an alternative 3' splice site. And later on we realized that if you look at the translational frames for these peptides, they actually correspond to the actual basic gene annotation. And we see that there is a new start site for this over here, which corresponds to this start of this gene here. And if you take a look at the amino acid sequence, we see that it could very well have been that there would have been a lysine over here, which resulted in this cleavage of this whole piece out. So we didn't observe this piece, the entire piece, because of the wrong enzyme. So we use trypsin in this case. Another piece of evidence that we get is the N-scan prediction, which is a gene prediction algorithm that tells us the possibility or likelihood of a particular segment to be expressed as a gene. So we see N-scan prediction for this entire region, which says that this is actually an intron. So what we've done is a targeted approach here to use a different enzyme to get this entire region. So instead of using trypsin, we can use Loci in this case, which will give us this entire, almost the entire length. And we can actually calculate the mass of this sequence and target them using a targeted MS approach. So once we do this, we can obtain the desired sequence. And what you see here is a bed track for this peptide sequence that we've identified based on our targeted approach using a different enzyme. So the second example I'd like to show you is the mapping of alternative new translational start site. So what's shown here is the beginning of the gene, where there is a 5' UTR here, and the data tracks above these are all map splice evidence. So these are all obtained by our RNA-seq data. So this is a bit of a complicated site where we actually look at multiple pieces of evidence that are coming from both genomics, transcriptomics, and our proteomics to find out if this is a correct assignment. So if you look at map splice evidence, where you see this line here shows that there is a possible exon on this end, and the exon actually ends here based on this map splice evidence. So if you zoom in on this region, which you see here, which actually is the 5' UTR, but our peptides that were assigned right outside of that for both these runs. If you closely look at the translational frame for these two peptides, for both peptides, it is in the same frame as this annotated gene, but it lies outside. So if you look at these potential start codons, the methionines here suggest that there could be several potential start codons for this new translational start site. Going back and looking at the map splice data track here, you can see that, well, this part seems like there is evidence for an exon, because this data track shows the connection between exons in map splice. So if you look at this in the zoomed in version, you can actually assign a new translational start site for this gene. And we found evidence for that new translational start site in the peptides. So the third example is where we found a peptide that spanned the exon-intron boundary in both runs. And this tells us that there could be a possible alternative 5' splice site downstream of this intron here, somewhere down here. But what was interesting is that we could also obtain quantitative data for this particular example with significant p-values. And in this case, we see a downregulation in the basal subtype compared to luminal. At this point, we are only able to do splice-site quantification, not at the isoform level. We are also doing, in some cases where we have multiple peptide level evidence, we are trying to develop multiple hypothesis testing methods to actually do this quantification at the isoform level, which I don't think I have time to go through. So the final example I like to show is a case where we found 3' splice site. And in this case, we see many peptides being assigned to this gene, some having exon skipping or exon-exon splicing. And what we are interested in is this peptide here. If you zoom in on this region, we can see that these lie really outside of this exon on the intron itself. And if you look at the evidence here for this peptide, it actually maps to this exon. So this could very well be an alternative 3' splice site over here on this intron, because we see this evidence at multiple runs, as well as in both luminal and basal subtypes of tumors. So overall, we've been able to identify 573 novel exon-exon splice sites. This doesn't really account for the intron-exon or exon-intron boundaries that I have gone through, just the novel exon-exon splice sites, which we have actually obtained evidence by looking to see if they were actually present in the UniProt databases, which we didn't. So we haven't stopped at that point where we do some sort of verification on these peptide hits by actually making synthetic peptides for these novel splice sites to the synthetic peptide. Currently, we are also doing RT-PCR to validate these splice junctions and to see if we can get quantitative information by using those types of assays. So finally, my personal outlook is that we can use various levels of data to actually classify breast cancer, not just use proteomics or gene expression arrays or the 5 methods that were shown earlier. We can actually improve the resolution of the subtypes classification by incorporating isoform level information or even our splice site level information. And we can also probably do that quantitatively right now. We can also expand this to the other subtypes and get classification maps that I've shown you earlier. And in the future, we could probably do larger cohorts of patient samples and classify breast cancer in a meaningful way. With the improvement of the technology, especially Orbitrap technologies, which could even become more sensitive and faster, we hope that we could even achieve greater depth in our analysis and at a faster pace, because all this analysis takes up a lot of time, informatics especially, but the analysis itself took about a month for us to do just running the experiments. So with that, I would like to summarize our experiments that we've done so far for global quantitative proteomics. We've identified a large number of proteins. This is really important because if you're doing proteogenomics, you need to have good data sets to look at things that are low and things that cannot be assigned to the conventional UniProt databases. Differential expression ratios are 8,500 or more. The label-free quantification is in agreement with for the limited RPPA data sets that we have, and also the spiking SILAC data that we have. We've also got iTrack data from other members of the network to also look at our data, and they are in very close agreement. The targeted quantitative discovery proteomics experiments looking at spliceosome, we've been able to achieve a large number of spliceosomal components, about 120 of the 150 or so annotated proteins. New trans factors and splicing factors have shown to be differentially expressed between normal and breast cancer, and also between the breast cancer subtypes. Our proteogenomic experiments have identified over 550 novel exon-exon junctions. In some cases, we are able to do the quantification, and currently we are doing validation experiments and also verification experiments by using spiking standards. So with that, I would like to acknowledge all the contributors to this work, collaborators, as well as the funding agencies, and especially thank the organizers for inviting me here to give this talk. And thank you all for your attention. And If you have any questions, I'll be happy to answer them.